Hello, my name is John Pandolfino. I'm the current president of the ANMS, and I would like to welcome everyone tonight to the ANMS Scientific Virtual Symposium. It's also my honor to introduce our moderator tonight, Dr. Fievos Christofi. Dr. Christofi is a tenured professor in the Department of Anesthesiology, Physiology, and Cell Biology at the Wexner Medical Center at The Ohio State University. Fievos has been really very active in the ANMS throughout his years, serving previously as a counselor, and now he is currently our treasurer. For those of you who don't know Fievos, he's also a very big and prominent person in the world of neurogastroenterology. And one of the biggest things that I think that people recognize him for is his contribution as the basic science editor for NGM. But he's really been a prolific researcher in many aspects of neurogastroenterology and has been funded for over 20 years. And I really can't think of a better moderator for this session focused on basic concepts in neurogastroenterology and specifically focusing on the relationship of GI dysfunction to central neurologic disorders. So without further delay, Fievos, take it away.
Thank you, John, for that kind introduction. It's really an honor to moderate this great virtual symposium. We have three amazing speakers today. The talks are 15 minutes each with 15 minutes Q&A at the end. And I encourage you to submit your questions through the Q&A box, and I will do my best to ask each of your questions at the end as time permits. It's my privilege now to introduce our first speaker. Dr. Alberto Travagli is a professor in neuro and behavioral sciences at Penn State University. His research is focused on the neurophysiological mechanisms underlying the central control of GI motility and changes that occur as a consequence of Parkinson's disease, stress, and other behaviors. Alberto is well funded by NIH and other agencies, and today he will talk to us about pathogenic mechanisms involving basal ganglia and Nigro-Vagal neurocircuitry linked to GI dysmotility and Parkinsonism. Alberto?
Slideshow, come on. All right. Well, thank you, Fievos, for the nice introduction. And I would also like to thank the organizers to give me the opportunity to present the data that I've generated in my laboratory at Penn State. And is going to deal with the gastrointestinal dysmotilities in Parkinson's disease and their relation with the neural circuits. As everybody knows, Parkinson's disease is a motor disease that is determined by the degeneration of dopaminergic fibers from the substantia nigra pars compacta onto the dorsal striatum via either indirect or direct pathway. The overall outcome of this deficit in dopaminergic inputs is a decrease of the inputs from the thalamus to the motor cortex, which then induce bradykinesia, tremor arrest, rigidity, and postural instability. But Parkinson's disease is also recognized, as initially described by Dr. Parkinson in his essay on the shaking palsy, as having other non-motor diseases, including GI dysfunction that referred to as dysphagia, esophageal and gastric dysmotility, delayed gastric emptying, nausea, and severe constipation. Interestingly, these GI and other dysfunctions are prodromal to the motor symptoms, and they may occur, at least for the gastrointestinal dysfunction, up to 15 years prior to the motor dysfunctions. So the reason of this connection between the substantia nigra pars compacta and the GI tract might be the anatomical connection that we have described recently that connects the substantia nigra pars compacta to the dorsal motor nucleus of the vagus and then to the GI tract via this Nigro-Vagal pathway. The Nigro-Vagal pathway that we have described recently, interestingly, provides an input to the stomach And we can see that if we stimulate with microinjection of an MDA in the substantia nigra pars compacta, if we stimulate the pars compacta inputs onto the dorsal motor nucleus of the vagus, we have an increase in motility and in tone of the corpus and the antrum too. In this case, I'm showing the traces from the corpus. And when this stimulation is preceded by a subdiaphragmatic vagotomy, injection of an MDA in the substantia nigra pars compacta do not induce anymore any response at the level of the stomach. Again, this is just the corpus, but we are seeing the same thing happening in the antrum. If animals are pre-treated with 6-hydroxydopamine, which is a neurotoxin that destroys the dopaminergic neurons of the pars compacta, we see that injection of an MDA does not induce any effect into the level of the stomach. This data indicate that the stimulation of the pars compacta works through a vagal pathways to control the gastrointestinal, to modulate the gastrointestinal motility. This pathway we also show that is tonically active, because if we put in the inhibitory allorodopsins in the pars compacta and then we shine the laser at the level of the dorsal motor nucleus of the vagus, we have a decrease in tone and motility, which is reversible upon switching off the laser light. And we can also reduce, by shining the green light, we can also reduce the effect obtained upon microinjection of an MDA in the substantia nigra pars compacta. The same type of results we are obtaining it when we stimulate the pars compacta while recording from the circular muscle of the proximal colon. And in fact, we can see over here that when we microinject an MDA in the pars compacta, we have an increase in both the tone as well as the motility of the colon. And again, this one is a vagally mediated effect, because if we conduct a subdiaphragmatic vagotomy, a microinjection of an MDA does not induce any response at the level of the proximal colon. Now, the question that have been interested many of the people that study the etiology of Parkinson's disease is whether the Parkinson starts from the bottom and then travels all the way up to the central nervous system, or starts from the central nervous system and goes all the way down to affect gastrointestinal motility, among other issues. There are valid arguments that support both a top-down as well as a bottom-up kind of etiology, but we have to consider that these two pathways, or these two occurrences, are not mutually exclusive, and most likely there is a combination of both pathways that are involved in this etiology of Parkinson's disease. For the topic of tonight's talk, I'm going to talk about the bottom-up. And this bottom-up hypothesis started with the work of a German pathologist, Brack, that a few years back put forward the hypothesis that there is an entry of neurotopic pathogens that goes into the GI tract. He also later developed it as going over through the olfactory bulbs and the olfactory pathway, but it's a different topic. So once this neurotopic pathogen enters into the GI tract, is ingested into the GI tract, it infiltrates the enteric nervous system, induces the misfolding of alpha-synuclein, which then travels through vagal pathway in a retrograde fashion all the way up to the central nervous system, where it reaches the first nucleus of the dorsal motor nucleus of the vagus, and then from there spreads up to the various nuclei that are involved. the connection again between the dorsal motor nucleus of the vagus and the pars compacta has been shown, that is via this Nigro-Vagal pathway that we have described recently. So we wanted to use an environmental model of Parkinsonism so that we could have somehow repropose this environmental pathogen that enters into the GI tract. And to do so, we use methods in which we are giving oral administration of subthreshold doses of the herbicide paraquat, which is well recognized to induce Parkinson's disease at higher doses up to 100 milligrams per kilo. And we combine it with lectin that in this case would have worked as a chaperone. And in fact, lectins are developed to deliver nanoparticles to area otherwise very cumbersome to reach. So when we give oral administration of paraquat and lectin, we obtain some sort of Parkinsonian effect. And we can see here in the same animal that we can do a very simple motor test. It's called the Vibrisse test. And so when the animal touches with his Vibrisse the bench, it just spreads out the paw to prevent hitting the bench. So in initial conditions, I would love to be able to do this. Let me see if I can. How do I do this? Here it is. No. In initial conditions, when we put the animal toward the bench, it easily projects the forepaw to prevent hitting the bench. After treatment with paraquat plus lectin, this movement is not available, is not smooth as it was before, indicating that there was the motor functions were compromised. But we were able to recover the motor function if we treat the same animal with a combination of levodopa and benzerazide, which is the typical treatment of Parkinson. And this effect of paraquat plus lectin was prevented if we conduct a preventative subdiaphragmatic vagotomy in the animal prior to giving the P plus cell treatment. We will then look at the histological landmarks of Parkinson's disease, and we can see in the upper portion the substantia nigra pars compacta in a control animal, where in brown we can see the tyrosine hydroxylase immunoreactivity, and after treatment with paraquat plus lectin at the same level of the pars compacta, there is a much lower expression of tyrosine hydroxylase, indicating that there was dopaminergic degeneration. We then looked at misfolded alpha-synuclein, which is the hallmark of Parkinson's disease. And we can see that in controlling the enteric nervous system of stomach, small or large intestine, there is no misfolded alpha-synuclein, which instead was present in the enteric nervous system of animals that were treated with paraquat and lectin. Misfolded alpha-synuclein was also observed in blue in the dorsal motor nucleus of the vagus of P plus cell treated animal, as well as in the substantia nigra pars compacta of P plus cell treated animals. However, if before doing the paraquat and lectin treatment we perform a subdiaphragmatic vagotomy, the misfolded alpha-synuclein is not present at the level of the dorsal motor nucleus of the vagus, but instead it's present in the cytoplasm of enteric neurons. So this model sort of recapitulates the hallmark histological and movement type of disorder that are seen in Parkinsonism. When we looked at the gastric emptying and the gastrocecal transit, we use the breath test with a 13C octanoic acid to measure gastric emptying, or with lactose ureide to measure gastrocecal transit. And we can see over here that in control and in P plus cell treated animal, there was a delay in gastric emptying as well as a delay in the gastrocecal transit in the P plus cell treated animal, indicating that, again, this P plus cell treatment was able to induce Parkinsonism with gastrointestinal dysfunction issues. So now the question was, how can we somehow translate or make some sort of speculation about what is occurring at the level of the animal when we are looking at the Nigro-Vagal pathway? And we thought that there was some sort of maladaptive plasticity going on. And so what we thought was that when Parkinsonism starts, or when Parkinson's disease starts, there is a delay in gastric emptying, there is a delay in gastrocecal transit, which induce constipation and which induce gastroparesis and constipation, respectively. So it could be that this reduced motility that is occurring at the level of the gastrointestinal tract is perceived somehow by the sensory inputs of the vagus nerve, which then transmits to the nucleus of the tractus solitarius at the level of the brainstem the information and tells the nucleus of the tractus solitarius, there is something wrong going on down in the GI tract. Can you do anything? And then the tractus solitarius sends the information to the excitatory inputs that are coming down to the dorsal motor nucleus of the vagus, including the substantia nigra pars compacta. However, in doing so, it puts oxidative stress along the Nigro-Vagal pathway, and this way sort of accelerates the transfer of the alpha-synucleopathy from the vagus nerve, from the GI tract all the way to the DMV, and through the Nigro-Vagal pathway to the substantia nigra pars compacta. So if this is true, if we are able to somehow impair or somehow reduce the activity of the Nigro-Vagal pathway, in theory, we should be able to reduce or prevent the spreading of this alpha-synucleopathy. To do so, we put in the inhibitory HM4-DI in the substantia nigra pars compacta that is in the DIO configuration, and so is not expressed, and it is picked up by cells that have an H-syn promoter. At the same time, we also put at the level of the dorsal motor nucleus of the vagus the retrograde tracer WGA that contains the CRE, so that whenever there are inputs that go from the pars compacta to the DMV, they get the CRE, and so the HM4 is switched and it's now operational, and it responds to external stimuli that can be given with either CNO or with the JH2 compound. So when we do this, we wait the appropriate amount of time, then we put CNO in the drinking water prior to the P plus cell gavage, and we wait. CNO will inhibit only the pathways, in this case, the substantia nigra pars compacta to the dorsal motor nucleus of the vagus pathway. If this one is correct, then we should have signs of reduced gastrointestinal issues, as well as reduced motility issues, motility in terms of movement disorders. And in fact, if we do unilateral chemogenetic inhibition of the Nigro-Vagal pathway, we have hemiparkinsonism, where the right paw, which is the pathway that has been protected by this inhibition of the Nigro-Vagal pathway, is normal, while instead the left paw shows signs of Parkinsonism. If we also recall this pathway, we can go and study and do recordings of the subthalamic nucleus, which is, if it is unprotected, it will start firing more because there is a decrease of the inhibition that goes from the external portion, the globus pallidus, to the STN, subthalamic nucleus. And in fact, in the unprotected side, we have an increase in the firing rate of the subthalamic nucleus, while instead the protected side, in which the Nigro-Vagal pathway has been inhibited, the firing of the subthalamic nucleus is normal. When we go and look at the bilateral chemogenetic inhibition of the Nigro-Vagal pathway, and we treat them with P+L, we see that the delayed gastrocecal transit, represented here in red, is attenuated by the inhibition of the Nigro-Vagal pathway that we obtain by treating the animal with JHU, starting prior to the P+L treatment. So in summary, we can say that gastrointestinal issues in Parkinsonian patients are prodromal to the motor issues, and the Nigro-Vagal pathway appears to modulate both the tone as well as the motility of the stomach and the proximal colon via vagal pathway. So gastric administration of subthreshold doses of paraquat together with lectins induce Parkinsonism, which is ameliorated by levodopa and is prevented by inhibition of the Nigro-Vagal pathway. In this model, we also have the misfolding of alpha-synuclein along the extent of the brain-gut axis, and ipsilateral blockade of the Nigro-Vagal pathway prevents the ascending synucleopathy induced by P+L treatment, while bilateral blockade of the Nigro-Vagal pathway prevents the delay of the gastrocecal transit time. And I would like to thank NIH for the support of this work, as well as the Michael J. Fox Foundation, the American Physiological Society, and the National Parkinson Foundation. And especially I want to recognize the people that have worked with this. This is a pre-COVID group, but Holly and Cameron are still working on this project. And previous project fellows are Luca and Georgia that went back to Italy, Michael Skolka that is now doing his residency in neurology at the Mayo Clinic, Laura Anselmi, Cecilia Bove that are working in other places as of now, and Jessica that is now a medical student at Stanford. The two neurologists that I work with at Penn State, Sub and Kala, and Michael Camilleri that does not have the need of being introduced by anybody because he's a giant in the field. Anhar Hassan, which is a neurologist at the Mayo Clinic, and Roberto De Giorgio that is working in Italy. And thank you for your attention, and I will be more than happy to take questions.
Thank you, Alberto. That was a fascinating talk. Please remember to submit your questions through the Q&A box. And now it's my distinct pleasure to introduce our next speaker, Dr. Kara Margolis. Is an associate professor of pediatrics at Columbia University Medical Center. She's a physician scientist that focuses on the role of the ENS in brain-gut axis disorders. She's funded by grants from NIH and DoD to conduct all her work. Kara is the director of the Pediatric Scientist Development Program at Columbia and associate director of the Columbia Digestive Disease and Liver Research Center. Today, Kara will talk to us about autism spectrum disorders and gut dysfunction and potential links with serotonin. Kara?
Thank you, Fievos, for those kind words, and I'd like to thank the organizers for inviting me to give this talk. Let's go on here. Okay. So, as Fievos said, I'm going to talk a little bit today about potential links with serotonin between autism spectrum disorders and gut dysfunction. So for those of you who don't know, autism is classically defined as a neurobehavioral diagnosis consisting of deficiencies in language and social interactions and accompanied by repetitive behaviors. However, it's been increasingly recognized that autism is an incredibly complex condition accompanied by many medical comorbidities, and GI problems are among the most common of these medical comorbidities. So GI problems are not only common in autism, but they're also found to aggravate both the core diagnostic symptoms of autism as well as problematic behavioral symptoms like self-injury and aggression. So these factors led us to believe could subsets of individuals with autism, instead of having a brain-centric disease, potentially have brain-gut axis disease. So gut-brain axis communication has been increasingly studied from the perspective of the peripheral systems. And peripheral serotonin is one of the systems that's been studied most frequently. I'm going to focus on peripheral serotonin today, but as you can see, the immune system and bacterial molecules are also important. And a lot of what I'm going to talk about that's available in the data shows peripheral serotonin interacting with the immune system and/or the gut microbiota. So it makes sense that serotonin could be a gut-brain microbiome link in autism. We know that serotonin is present in both the brain and the gut. However, although most of the research done in serotonin is in the brain, 95% of the body's serotonin is located in the gut. However, that 3% in the brain plays critical functions, both in central nervous system development as well as in the modulation of autism-associated behaviors. Similarly, gut serotonin plays critical roles in enteric nervous system development, as well as gastrointestinal motility. And gastrointestinal motility disorders tend to be the most common GI disorders found in individuals with autism. Similarly, there's been increasing data that's demonstrated the bidirectional relationship between serotonin and the gut microbiota, whereby serotonin modulation in the gut, say by like SSRI administration, can alter the type of gut microbiota that exists. And similarly, there's specific types of gut microbiota that can influence both the secretion and the production of gut serotonin. Similarly, 30% of individuals with autism have hyperserotonemia or high blood serotonin levels. And physiologically, it makes sense that that extra serotonin comes from the gut. In line with this, we found that hyperserotonemia was associated with both constipation and abdominal pain in children with autism. The first clinical study that links serotonin with functional gastrointestinal disease in autism was a study put out by Baylor a couple of years ago. And the group at Baylor, led by Ruth Ann Luna, studied 3 particular cohorts. They studied 2 neurotypical cohorts, one with functional GI disease and the other without. And the third cohort was one of children with ASD and functional GI disease. This is a principal component analysis. So every shape on that screen represents an entire bacterial community for a single individual. And what you can see by looking at this is that amongst the neurotypical populations, there was really no major difference in the gut microbiota communities between those neurotypical kids who had a functional GI disease and those who did not. In contrast, the children with autism and a functional GI disease had a quite different gut microbiota composition. And when they look further into this microbiota composition, you can see that there were several bacterial species that seem to be abnormally regulated. I point out Clostridia because there's been a number of studies that show differing levels of Clostridia in individuals with autism, and has also been shown as by Pernikasia, this relationship between Clostridia and serotonin. What they found was an association with these abnormalities in dysbiosis. They found an increase in serotonin as well as its building blocks, and also an increase in pro-inflammatory cytokines. And all of these associations were linked specifically with the kids with autism and functional GI disease. So what I'm going to show today is based on this study and how serotonin is related to autism. I'm going to show clinical data to make clinical correlations and show mouse models that will help elucidate potentially some of the mechanisms by which serotonin is working. I'm going to focus it as proof of concept on one genetic mutation, the SIRDALA56 mutation, which we've studied in our laboratory, and one environmental exposure, which is maternal infection and/or inflammation. So the SIRT-ALA56 mutation was based on the serotonin transporter, abbreviated here as SIRT, and SIRT variants have been found to be overexpressed in multiple studies of children with autism. Our collaborators, Dr. Blakely and Veenstra van der Wiel, who were at Vanderbilt at the time, conducted a genome-wide association study for SIRT-associated genetic abnormalities in autism. They identified several SIRT coding variants as risk factors in autism and took the most common coding variant, which was a glycine substitution for an alanine on the SIRT locus, and created the SIRT-ALA56 mouse. They found these mice not only had core autism-related behavioral abnormalities, but also serotonin-related brain abnormalities and hyperserotonemia. Because of this hyperserotonemia and the idea that serotonin and SIRT were also present in the gut, we formed the hypothesis that the SIRT-ALA56 mutation could be a brain-gut link in autism. The first thing we did was to evaluate the enteric nervous system in these mice. The genetic mutation of SIRT here made SIRT hyper-efficient. So we hypothesized that there would be less serotonin available in the enteric nervous system. Because serotonin is neurogenic, we postulated that both total and late-born enteric neuronal subsets that serotonin influences would be deficient in the SIRT-ALA56 mice. And we saw that that was true. When we looked in both the ileum and colon, and you can see total neurons in green, and an example of a late developing neuronal subset, the GABAergic neurons, here in blue, both were significantly deficient in the SIRT-ALA56 mice compared to wild type. And this translated into functional deficits in colonic motility. As you can see here, the SIRT-ALA56 mice had slower colonic motility than wild type mice. So they were constipated. Constipation happens to be the #1 GI problem in children with autism. We then went on by spatiotemporal mapping to analyze peristaltic contractions, which are primarily modulated by the enteric nervous system. And you can see here that each peristaltic contraction is represented by a single arrowhead. And just by looking at these contractions and comparing between the two groups of mice, you can see that peristaltic contractions were slower, less frequent, and shorter in the SIRT-ALA56 mice compared to wild type. And what this told us was that the slower colonic motility, or the constipation in these mice, was likely secondary to defects in the enteric nervous system. So we then went on to determine whether we could bypass the effects of SIRT during development to prevent the gut defects in the SIRT-ALA56 mice. So as I showed you, SIRT-ALA56 mice had less neurons and slower colonic motility. We decided to focus on agonism of the serotonin 4 receptor because we and others have shown that agonism of this receptor increases both enteric neurogenesis and gastrointestinal motility. We further found in a clinical study that kids with autism and gastrointestinal issues have lower intestinal levels of the serotonin 4 receptor, and a recent study revealed that DNA methylation levels of the serotonin 4 promoter are lower in autism than in neurotypical kids, showing that these kids might not only have less of an intestinal serotonin 4 receptor, but it may be less efficient. In response to this, we administered precalipride, the selective serotonin 4 receptor agonist, during development to the SIRT-ALA56 mice, and found that agonism of the serotonin 4 receptor indeed prevented the abnormalities both in enteric nervous system development and gastrointestinal motility. We saw normalization of total and late developing neuronal subsets in the submucosal plexus and in the myenteric plexus. And this translated into a normalization of both total and colonic transit, as well as a normalization of peristaltic contraction frequency, velocity, and distance, really implying that the reason that the colonic motility was fixed was because we were able to normalize the enteric nervous system. When we went to look under the electron microscope at the microbiome here, and this is a picture from the small intestine, you can see just visually that there was a lot of bacteria in the small intestine. When we did a simple study, just by looking at the amount of ribosomal 16S in the small intestine and comparing it to that of the colon, we found that there was significantly more ribosomal 16S in the small intestine of the SIRT-ALA56 mice over the colon, showing that these mice had small bowel bacterial overgrowth, another more common manifestation in kids with autism. We then collaborated with the group at Baylor to do deep sequencing of the microbiota. And what you see here is another principal component analysis, like I showed earlier. And what you can see, if you compare the SIRT-ALA56 mice in green who received saline compared to the wild type mice in red, you can see that their bacterial communities are quite distinct. However, when we gave the SIRT-ALA56 mice prucalopride throughout development, there was a normalization, or at least a partial normalization of their gut microbiota. We then did integrative analytics that indicated relationships between these enteric microbiota differences and gastrointestinal dysfunction, as well as behaviors such as social dominance. And these brain and behavioral outcomes are still being intensely studied. So what I've shown so far is that modulating serotonin, at least through the 5-HT4 receptor, SIRT, or the gut microbiome, may be viable brain-gut targets for autism. In the second part of this, I'm going to focus on an environmental influence, which is maternal inflammation and/or maternal infection, because both have been shown to significantly increase autism risk in clinical studies in offspring. Rodent models of maternal immune activation, which I have abbreviated as MIA, have reliably mimicked the model of maternal infection. And this is often done by prenatal administration of a viral polymimetic called poly-IC. Mice that receive poly-IC develop core autism symptomatology and neuropathology consistent with autism. Interestingly, however, they also have placentas with very high serotonin levels that are associated with increased pro-inflammatory cytokines and abnormalities in hippocampal SIRT levels. Based on this, several investigators went to look at how serotonin, the immune system, and the microbiome might be linked to both the gut and brain in maternal infection in autism. One of the nicer studies that came out of this was from Sarkis Mazmanian's group, where they showed that maternal immune activation leads to a dysbiosis of the gut microbiota that's driven primarily by alterations in Clostridia and Bacteroidea. This was associated with diminished epithelial barrier integrity and an altered serum metabolome, including not only increased levels of serotonin, but also levels of indole pyruvate, which is a key molecule in tryptophan metabolism. These altered molecules then leak through this epithelial barrier to cause detrimental effects on the brain and behavior. In the next set of studies, the investigators repleted the mice with Bacteroides fragilis, and they chose this not only because it was abnormal in the MIA model, but because it had been found in clinical studies to be selectively depleted in some individuals with autism. What they found after repletion with Bacteroides was that there was a partial normalization of the microbiota, improved epithelial barrier integrity, and restored serum metabolites. And here I show the normalized levels of indole pyruvate, as well as reduced leakage of the GI metabolites because of the improved barrier integrity. And this led to beneficial effects on brain and behavior, including in stereotypies, communication, and social interaction. So what I wanted to highlight here was in this rodent model of maternal immune activation, this relationship between placenta and high serotonin levels, because I think this is where a lot of the field is going. The question of could the developmental impact of serotonin be modulated by the placenta? And I'm going to show you how in our two models that's now relevant. On the left, you have the SIRT-ALA56 mice, and on the right, the maternal immune activation mice. Placentas are both affected by either low or high levels of serotonin, respectively, in either model. But serotonin in either direction leads to an immune activation. These low and high serotonin levels then persist and go to the brain, whereas the SIRT-ALA56 mice have low brain serotonin levels and the MIA mice high serotonin levels. This leads to abnormalities in central nervous system development that also are consistent with behaviors seen in autism. Through a set of other studies, it's been shown that there's also less and more serotonin and/or serotonin signaling in these mice, and that leads to abnormalities in enteric nervous system development, gastrointestinal function, and/or the gastrointestinal microbiota, really showing that developmental serotonin may impact the brain, gut, and microbiome abnormalities consistent with autism-associated phenotypes, and leads to the question as to whether microbial and/or serotonin-based therapies may be helpful in these conditions. So in the future, I'm just going to review one quick study that we're doing right now. There's always a lot of questions about serotonin and the microbiome, and that could be a whole other 15-minute conversation within itself. But I thought this phylogenetic tree really symbolizes nicely that there's no one microbiome for autism. This tree is a compilation of a number of studies done over a 10 to 15 year period. And what you can see literally is that the microbiome in these patients is all over the map. The next step, however, is to look into see which specific microbiota or metabolomics in actual humans impacts patients with these different complex conditions. And so what we've done as part of a multi-center study that's been spearheaded, again, by Dr. Luna at Texas Children's Hospital, is we've recruited over 400 children, half with autism, and did comprehensive phenotypic assessments, not only of their behavior and gastrointestinal tracts, but also their nutrition and their active medical issues. And like I said, these children have a lot of medical comorbidities, which very well may influence their gut-brain axis. From the stool samples, we've looked at the microbiome and metabolome, and we've now conducted multi-omic analysis to determine how specific parts of the microbiome and metabolome correlate with GI problems, behaviors, other medical issues, and nutrition. I'm the PI at the Center for Discovery site, which is a residential center in which kids receive carefully controlled diets as well as specific behavioral modifications. These results should be published within the next several months and will really lend key insights into what may be contributing to some of the diversity in the microbiome and metabolome here. So in conclusion, GI issues are common in autism, and serotonin may play an important role in the development and/or perpetuation of the gut and brain abnormalities in autism. So targeting serotonin may ultimately be a viable therapeutic option, but the mechanisms still remain largely unknown, so a lot of research is warranted. Furthermore, there are a number of other good gut-brain examples in autism, and particularly the BTBR model also shows abnormalities in serotonin in the gut. But I don't have time to focus on the other models, so I just mentioned them here. So quickly, I'd just like to make note, I've highlighted my key collaborators for the data I've presented here, but really the slide is just to emphasize that none of my research, both basic and clinical, goes on without true collaborations, for which I'm very fortunate, as well as my research support. And also I'd like to thank the Center for Discovery, which is where I conduct the clinical studies, including the one I mentioned at the end here. Thank you.
Thank you, Kara, for your talk. Very impressive as always. Just a reminder, you can submit additional questions for our speakers through the Q&A box. We already have quite a few questions, so thank you for doing that. I'm delighted to introduce our final speaker, Dr. Helen Raybould. She's a distinguished professor in the Department of Anatomy, Physiology, and Cell Biology at UC Davis School of Veterinary Medicine. She has done pioneering work on the vagal gut-brain connection and how changes in the composition of gut luminal contents, such as nutrients and bacterial metabolites, influence gut function, appetite, and obesity. Today, Helen is talking about how bacterial metabolites influence the vagal afferent pathway in appetite regulation and in obesity. Helen?
Thank you very much for that introduction, Fievos, and thanks for the organizers for inviting me to talk today. So I'm going to talk about some work that we have been doing in my lab, and I feel a little bit like the orphan child because I'm not really talking about central nervous system pathways, but I'm really talking about bacterial metabolites in the vagal afferent pathway. But I guess we can consider obesity as a disorder of the central nervous system in terms of changes in the input from the gut in response to obesity. Okay. I have to work out how to advance my slides. There we go. So we know that the gut microbiota has many beneficial effects, and it can regulate mucosal immunity in the gut, barrier integrity, and overall gut health. But the important thing is that it also has effect on the host beyond the gut. So obesity, type 2 diabetes, and a number of different neurological diseases, function and pathology have been associated with changes in the gut microbiota. The key thing that we're struggling with right now, I think, in this field is really to move from these descriptive studies to really understand cause and the causality of the microbiota. And in order to do that, we've turned our attention, as have other groups, to looking at the microbial metabolites, at one way in which these microbes will be communicating with the host. So the microbes make these metabolites. Basically, they use substrates that are in the lumen of the gut, and this can be from the diet of the individual. So they predominantly use complex carbohydrates, so fibers, complex oligosaccharides that are in plant material and milk, but are also in the intestinal mucus. And they can also use residual amino acids and proteins that are undigested and absorbed. There are several classes of microbial metabolites, and the ones I'm going to talk about today are short-chain fatty acids and the tryptophan derivatives, but this is by no means a complete list. So this is a sort of summary diagram from a nice review by Fetisov from 2016. So here you can see the luminal environment with the microbes and the epithelium. And these microbes can use, as I said, the partially digested nutrients and the fibers for their own metabolic needs, and then produce a number of different substrates that are then in the lumen of the gut and that can have an effect on the host. So there are substrates like lactate and butyrate, short-chain fatty acids. There are also other bioactive molecules, as we've heard the previous speaker talk about some of the tryptophan derivatives like 5-hydroxytryptamine and also the indole derivatives. And then there are many other classes of metabolites that these bacteria make, but essentially they can either have an effect at the level of the gut epithelium or the gut immune cells. They can also stimulate hormones such as satiety hormones from endocrine cells. They may be able to influence enteric neurons. What I'm going to talk about today is the effect that they have on vagal afferents. So short chain fatty acids are products of gut fermentation of dietary fiber. The three principal short chain fatty acids are acetate, propionate, and butyrate. Acetate is produced by most gut microbes and is at the highest level of any of these short-chain fatty acids. Propionate mainly comes from Lactospiraceae, and butyrate's widely distributed in the gram-positive firmicutes. Now, after production in the gut lumen, these short-chain fatty acids are rapidly absorbed by specific transporters that are expressed by colonocytes and are used by the colonocytes for metabolism. So one must interpret fecal or colonic levels with caution. Most of these are at fairly low levels in the plasma, and this has been done in very good isotopic tracer studies. So this begs the question as to whether they really are acting predominantly within the gut wall rather than systemically or in other ways. So there was a very nice study in 2016 that came out that showed that colonic propionate in humans changed anticipatory reward in the human striatum and altered food intake. And they really did this in a nice way. They conjugated propionate to inulin, a fiber. And so the propionate would not be released from this complex until it got to the colon. And basically what they found, if they looked in the striatum, and this was specifically in the caudate region, they found that the activation as measured by the bold signal in individuals on high energy diets was decreased by administration of propionate. They also found that energy intake went down, and this was correlated with the amount of propionate that was taken in. So this really does suggest that this may be very relevant in humans as well. There have been a few studies in animal models, and this is again from a review that came out quite recently. Acetate has been shown to have effects in the brain to change food intake. It also has effect on other metabolic tissues like fat. It has effects on the pancreas and also the liver. But within the gut, it's been shown that acetate, butyrate, and propionate can all have effects via a specific G protein coupled receptors expressed by endocrine cells that can then have an effect either through humoral pathways or through neural pathways. I just want to highlight one study that came out a couple of years ago where they gave butyrate to high-fat fed mice, and they found that butyrate would decrease food intake and also decrease the high-fat diet-induced increase in body weight. This was abolished by vagotomy, suggesting that it could be mediated through a pathway. And there's another study that has also demonstrated the role of the vagal pathway. So we looked at the effects of butyrate in high-fat fed mice, and we basically found that there was an improvement in permeability in these mice when we gave them butyrate. There is a decrease in colonic inflammation. In this case, we measured it by measuring lepicalin-2 in the feces, although we also looked at expression of various markers by PCR in the wall of different regions of the gut. So one of the things that my lab has been interested in is sort of the effects of high-fat diets on the vagal afferent pathway and sort of the interrelationship between changes in gut permeability and inflammation and how this may alter vagal afferents, the role of the gut microbiota, and bacterial products that have deleterious effects like lipopolysaccharide that can attenuate the effect of various luminal substances on vagal afferents, and how this can then lead to hyperphagia and obesity. So what we did in these animals is that we measured the inhibition of motility of food intake in response to CCK. And as you can see in low-fat fed animals, you get a robust inhibition of food intake in response to CCK. This is when we gave butyrate to these low-fat fed animals, there's no difference. In high-fat fed animals, you get this attenuation of the CCK response. And this was completely restored back to low-fat fed controls when we gave butyrate to these high-fat fed animals. So we're doing something to this gut-brain axis. And we don't think that it's changes in the gut microbiota. Our hypothesis is that it's really a direct effect of butyrate at the level of the terminals of the vagal afferent neurons in the gut wall. So short chain fatty acids are certainly very interesting molecules, and there's a lot more that we need to do. We've also been looking at tryptophan metabolism and tryptophan derivatives made by the bacteria. So as we heard again in the prior talk, that tryptophan can be used by the host, both in the gut and in the brain, to produce neurotransmitters like serotonin. But it can also be used by the gut microbiota. And the gut microbiota can use tryptophan and it can make indole derivatives. One of the ones we're interested in is indole-3 lactate. And in addition, the host can also use tryptophan to make kynurenine and kynurenine metabolites. And we've been interested in one of these, kynurenic acid. Now, I'm very fortunate at UC Davis. I work alongside a very talented glycochemist called Dani Beria. And what she can do is to provide us with very specific kinds of fiber that's present in human milk. And these are these human milk oligosaccharides, which, as you can see, this is the constituents of milk. Here's lactose, proteins. And you can see that oligosaccharides make up a large proportion of what's present in human milk. They're abundant. We know that they're selective prebiotics, and that in and of themselves, they can have beneficial effects. But why they're there probably is that the host can't use them. So the infant can't use these oligosaccharides. We don't have the enzymes to break them down, but the bacteria do. So probiotic bacteria like bifidobacteria can break down these oligosaccharides and produce different metabolites. There's lots of different oligosaccharides. Danny can provide us with pure chemically characterized samples of LNT, lacto-N-triose, 6-xylolactose, and 2-fucosylactose, which we have had in sufficient quantities to be able to do some in vivo as well as some in vitro experiments. So we gave human milk oligosaccharides to high-fat and low-fed animals. And basically, we saw the same thing as we did with butyrate, that looking at both inhibition of food intake in response to CCK, we see, again, as I mentioned before, you see this robust inhibition of food intake in response to CCK. It's abolished in high-fat fed animals, and we can restore it by giving these high-fat fed mice these human milk oligosaccharides. In this case, it was 2-fucosylactose. We can also restore the FOS response. So here you can see the sections of the brainstem. FOS is a marker for activated neurons. Here's the area postrema, here's the NTS, these regions where these vagal afferents terminate. And you can see robust activation of FOS in response to CCK in low-fat fed mice, which is attenuated in high-fat fed mice. And this is restored in mice when we give them 2-fucosylactose. And here's the quantification of this response. So this is really pretty exciting because it shows that giving these human milk oligosaccharides can actually have beneficial effects on this gut-brain axis. So there's a number of ways in which these oligosaccharides may be acting. They may be having direct effects, or they may actually be having their effects by changing the gut microbiota. We also work alongside a microbiologist, David Mills at UC Davis, and he works on bifidobacteria. And what he has found is that there are some species of bifidobacteria that have the ability to use these very specific milk oligosaccharides. So these are infant-born bifidobacteria that have been isolated from infant poop. And what characterizes these bifidobacteria is that they express very specific transporters that can take these human milk oligosaccharides into the bacterial cell. They also express specific glycosidases that can then break down these complex oligosaccharides, and it provides energy for the bacteria. So it really gives them an advantage against other bacteria that are present in the infant gut because they express and have the ability to use this predominant substrate that's sitting there for them from human milk. But what they do, other than use this for energy, is that they then spit out a number of other metabolites. And so there's an efflux mechanism by which these metabolites are then spat out of the bacterial cell into the lumen of the gut. And one of these that we've been interested in is indole-3 lactate. So we had the opportunity to look at infant stool samples, and we characterized a number of different stool samples looking at 16S abundance of bifidobacteria. And basically we categorized them into low bifidobacteria stool and high bifidobacteria stool. And you can see that in these stools, the green is all bifidobacteria. So these are infant stools, these are breastfed infants, and you can see these very high predominance of bifidobacteria. We then did metabolomics on these stool, and we found that there were some changes in short chain fatty acids like acetate and lactate when you had high BIF. But the one that really stuck out to us was indole-3 lactate. So there was a very high enrichment of indole-3 lactate in the poop that was high in bifidobacteria. So we then took one of these bifidobacteria, this is B. infantis, one strain of B. infantis. We grew it either on lactose and HMO, and then took the growth media and did metabolomics by NMR. And again, we found that when you grow the bugs on human milk oligos versus on lactose, you can enrich indole-3 lactate. The other thing that we did was we added tryptophan to the growth media, and we found a dose-dependent increase in the production of indole-3 lactate when we added increasing doses of tryptophan to the growth media. So what does indole-3 lactate do to the gut-brain axis? So we took nodose neurons. These are neurons that are innervating the stomach and the intestine. We took them, the cell bodies, we cultured them in short-term culture. Here's a nice pretty picture of these neurons. And then we looked at the responses to ILA, to indole-3 lactate. Now, as I'm sure many of you know, that there is evidence from my group and several other groups, and also from humans, for the attenuation of this vagal afferent pathway in obese animals and in animals fed high-fat diets. And so we want to understand why you get this attenuation, because we think this is what leads to hyperphagia and obesity. And it's obviously a very tractable target for obesity treatments. And one of the characteristics is that these neurons no longer respond to leptin and no longer respond to CCK. So we measure the response to leptin in these cultured neurons, and you can see a small phosphorylation of pStat3 in response to leptin in these vagal afferent neurons. And when we add indole-3 lactate, you can see we markedly upregulate the phosphorylation STAT3 in response to leptin. But this is not recapitulated when we use a similar molecule, indole. And this is also seen if we look at another marker of leptin activation, which is phosphorylation of ERK. Now, we don't actually know what receptor that ILA might be working on, but there is some evidence that it can act at the aryl hydrocarbon receptor, and so we're pursuing this. Now, we have begun to look at other metabolites. As I mentioned, we're really very interested in chymotryptic acid and the receptor that might be working on. And we can see similar beneficial effect of some of these other tryptophan metabolites on the vagal afferent neurons. So I would just like to finish now and thank you for your attention, and also to thank the people in my lab who actually have done the work. Particularly like to thank Michael Goodson and Danny Zumpano in my lab. Two prior members in my lab, Amy Ehrlich and KP, did a lot of work with the vagal afferent neurons, and Amy did the work with the ILA. And I'd also like to thank my collaborators at UC Davis, Danny Berea, Dave Mills, Carlito Labria, and Carolyn Slupsky, and also those who fund the work. Thanks very much.
Thank you, Helen, for your wonderful talk. We are running a little bit late, but I'm going to try and stick around and have a 15-minute Q&A period. And I'm hoping that most of you can stick around, as there are quite a few questions. So I'm going to try and get some of these questions answered. I'll ask the first question, and it has to do with, it's to Alberto. And this has to do with gastric motility and the effects of an NDA on vagally mediated gastric motility and the development of tone. The question has to do with regional differences in effects on motility. The effect of an NMDA on vagally mediated effects on gastric motility appears to be specific to the proximal stomach, because tone is not typical response of the distal stomach. So have you done differential measurements on the region-specific responses to the vagally mediated responses to NMDA?
No, that's the quick answer. The recording that I've shown were all made at the level of the proximal stomach. We put our strain gauges at the border between the fundus and the corpus, and we are recording the corpus motility, the corpus circular muscle. We have not done antrum, and so I cannot really do it. However, we have plenty.
Can you speculate on what you might see?
I don't like speculation. Not have no. If I were to have an n equal to 1, I would speculate for 15 minutes with no problem. But I do not have any data, just proximal corpus.
Okay, follow-up question to that. What is the nature contents of the meal that was used for gastric emptying studies?
We use a commercially available pancake. We fast the animals overnight, and when we are doing either the gastric emptying on the gastrocecal transit time, we give the fasted animal 1 gram of commercially available pancake, and with the octanoic acid or with the lactose ureide.
Okay. There was a question about serum biomarkers such as BDNF and their associations with GI symptoms in Parkinson's.
We have not done anything on with serum markers. No, sorry.
Okay. So so this is a question for Kara. The question has to do with, let's see, with serotonin levels. At what point in the development does the blood-brain barrier resist serotonin movement from the serum to the brain?
Sure, that's a great question. You know, I think the blood-brain barrier starts to form, I think, in mice around E12, and it becomes functional around that stage. But I don't know if we know the exact time when serotonin stops transferring through. In humans, tight junctions form at about 14 weeks gestation. So it forms pretty early. But I think that when you're looking at influence of serotonin, I guess the most applicable studies might look at like SSRI exposure in humans. So when you look at first trimester SSRI exposure, there's probably the highest rate of issues with long-term cognitive and brain neurodevelopment. However, when you look at total cumulative SSRI exposure throughout pregnancy, that's also an important predictor of neurodevelopmental outcomes. And so what that's telling us is that it may not be only about blood-brain barrier permeability to serotonin, but serotonin to get into the brain in those early stages and then have downstream effects.
I see. There was also a general question about serotonin. How are levels of serotonin controlled in our body? The safety margin seems to be so low.
Yeah. You know, I don't think this has been looked at a whole lot, but I think that, you know, certainly we've seen, at least in our mouse models, that when you alter serotonergic signaling pathways, either through enterochromaffin cell production or enteric nervous system production, that there's definitely pretty quick alterations, say in tryptophan hydroxylase levels in those areas, showing that the gut probably does play a role in regulating serotonin. The other thing that's important to consider when you're looking at serotonin levels is what platelet SIRT is like, because really the best marker for looking at blood serotonin levels is going to be found in the platelets. And so when you look at, you know, a SIRT knockout model, you're not going to be able to really make any judgment of serotonin levels in those mice because they don't have SIRT in the platelets, which don't produce serotonin, but actually just take it up. So it's a complicated question. And I think the point I was hopefully trying to make was that we're trying to figure out that balance, but we don't even yet know what these extremes exactly do and what mechanisms they work through to influence enteric nervous system development.
Thank you, Kara. I appreciate the complexity of the system. So the other question relates to your LF56 mice. What is the nature of the neuronal loss in the ENS? Does it involve motor neurons, sensory or interneurons? What do you know about that? And were tissue studies performed on colonic muscles to determine the nature of the neuromotor defects?
Right. So, you know, so a number of questions. I think the last question, no. But certainly I can probably guess who asked that question, and they're certainly welcome to look with me if they like. In terms, we didn't look at motor and sensory neurons. We did look at neuronal subtypes. We really focused on the neuronal subtypes that we found in earlier studies that serotonin had been shown to affect the development of, which focused on the late developing neuronal subtypes. So certainly that included like CGRP expressing neurons, which it did affect, but we haven't done a real thorough evaluation.
Have you looked at inhibitory motor neurons or?
So we did in other studies look at nitric oxide expressing neurons and found that there wasn't a significant difference. We have not looked at cholinergic neuron numbers in them.
Okay, okay, so it's work in progress.
It's work in progress, yes.
Okay, so next question I have here is for Helen. Helen, there is a variety of strategies from what you discussed and what you've done. There's a variety of strategies that are available to treat diet-induced obesity, as I understand it, by acting on vagal afferent signaling and firing to reduce food intake, etc. And you talked about human milk oligosaccharides as potential probiotic substances to stimulate indole 3 lactase. You've done work on leptin receptor signaling on vagal afferents, some studies on butyrate, other microbiome metabolites, and even some earlier studies, very elegant studies that you've done on herbal medicines and how they can impact obesity. Can you kind of put it into perspective for us? And you're probably in the best position to comment on this. Can you comment on the therapeutic potential of these strategies, and are any of these being or will be tested in clinical trials sometime in the future or hopefully soon? Like, for example, oral supplements of butyrate, which doesn't seem like a very difficult thing to do. So Helen?
Yeah, so I think the potential is there, certainly for being able to use some of these metabolites. I think that there's still, some of these have been shown to be effective in animal models. And then when they are taken to sort of human patients, it's hard to show effectiveness, particularly if you look at prebiotics and probiotics, particularly probiotics. And that's not only for obesity, but also in things like inflammatory bowel disease too, that there is this very difficult translational kind of blockage. And so one of the approaches, and I think it's because we don't understand mechanism, right? We don't really understand how the gut microbiota can be influencing the different components of this system. So I think that this is why sort of maybe looking at metabolites can be helpful. I think the one study that I talked about where they did the colonic propionate, what was nice about that study was that they used, as I mentioned, this inulin conjugated propionate. So rather than just give free butyrate or free propionate, they chemically attached it to inulin, and so that it wasn't released. And they actually showed that pretty nicely, that it wasn't released until it got down to the colon. And so it maybe needs to get to where it's normally made by the gut bacteria to have its effect. So I think there are lots of ways in which we can optimize, and make sure that it can be effective in humans. I just don't think we're there yet. But I think the promise is there for sure. And it may end up to be personalized, more of a precision thing, because it may depend on an individual's gut microbiota as to what they actually need too. So
yeah, I mean, there seems to be a lot of variability in studies on microbiota and the microbiome. So I think until we get a good handle on that, we perhaps with more standardized approaches, then we can pinpoint as Kara is doing and as you're doing, what more specific targets are that we can pursue in clinical trials. And I think one of your targets, I think in looking at all the studies, you may have a different opinion, but and please tell me, is the leptin signaling, I mean, it's been regulated in so many different fine ways, and that may be a target that could be pursued in terms of how different metabolites, microbiome metabolites, et cetera, how they interact with the leptin receptor signaling in the vagal afferents, and how that can be used in terms of interventions, et cetera.
Yep, right. Thank you.
So we are almost there. A couple of more minutes. I have one question for Kara and Alberto, and this has to do with translatability. I couldn't let you get away with not answering this question. When you look at Parkinson's disease and autism spectrum disorders, there is a lot of wonderful models that in some cases are acute, in some cases show differences in responsiveness and differences in mechanisms and so forth. Can you comment on, each of you, on the translatability of what we find in the animal models to the human disease? And Alberto, perhaps you can go first.
Ladies first. Come on, Fievos. We're Mediterraneans. We should do that. But Kara, please, if you want to go ahead.
No, it's fine. I think it's a great, it's an important question. And so I think rather than, looking I don't think when we look at animal models of autism, we're looking at autism. I think we have to look at specific phenotypes. And so certain things like sociability and repetitive behaviors have been shown in multiple studies to be phylogenetically conserved amongst the same serotonergic circuitry in both mice and humans. A lot of this has been obviously looked at much more extensively in the brain and the gut. We're starting to do that in the gut, and I haven't presented it because our data isn't complete yet, but we think that same conservation is happening there. So I think that when you're defining, I think even defining one autism is impossible. So bringing it to a mouse model is going to be even harder. But I think if we can look at specific phenotypes to figure out where we can drive serotonin pathways and where we can modulate this, I think that's going to be the better approach.
Okay. I think one of the things you mentioned, I think, in one of your slides was about the two models, the maternal immune activation MIA model, and then the LR5/6 model. What struck me was that in one model you have a decrease in 5-HT, and in the other model you have an increase, and both are associated with alterations in CNS development, as you would expect, but both are associated with autism spectrum disorder phenotypes, I guess, or some phenotype. So that's one question I had. Do we know which phenotypes, or is that something you're working on? Each of those models better represents?
No, it's the disadvantages of a 15-minute talk, or that there are different phenotypes associated with autism.
And I was intrigued by that, yeah.
That seemed to be, it's a good question, and it's something I just didn't have time to address. So yes, there are different phenotypes that are addressed in those ways. And it actually makes sense because when you translate it to the clinical population, you see that there are some patients with autism that do fantastically well on SSRIs, and there are some that just completely decompensate. And likely it has to do with this developmental circuitry and what these specific subsets need. But as Helen alluded to before with the microbiome, but I think it also goes with all of these other factors, is that personalized medicine and trying to figure out biomarkers for which patients are going to do well based on where these defects occur and when they occur is going to be really important.
Thank you, Kara. Alberto, you get the final word.
Wow. So translatability. Let's assume that what I show you is can be translated to humans. So if you take Parkinsonian patients at stage 1 at the very beginning, the issue with this is that you do not really know whether they are really Parkinsonian or if there is any other neurological problem in there. But if you take those and you follow them and you treat them with promotility drugs that works only in the periphery, they do not go through the brain, are you able to somehow delay the passage or the development of Parkinson? Perhaps going from stage 1 to stage 2 is going to take, let's say, 3 years. But if you give them a promotility agent, perhaps instead of 3 years, it's going to take 5 years to delay them. It's just an idea. It might be completely crazy, but I like crazy ideas, at least in the laboratory. In humans, I'm a little bit more skeptical and a little bit more careful. But I would say that if there is any validity in what I have shown, and I hope that there is, perhaps we are able to slow down the full bloom of Parkinson in patients if we treat them through the GI tract. Is it going to work in all of them? I don't think so, because again, top down or bottom up are two different things, but it would be worth giving a try.
Thank you, Alberto. Unfortunately, we're at the end of this great symposium. There's many more questions, but that's all we can get through. And thank you to everyone who stuck around to listen to our conversation. I want to thank all our speakers for their amazing talks. And the session is recorded and it will be posted on YouTube shortly. We can continue the conversation on Dark Matter. Please note that ANMS members are enrolled in Dark Matter. It's one of the benefits of membership. I want to thank you, thank everyone, and enjoy the rest of your day. Take care and stay safe.